So first, an overview of the HTB unit. In my opinion, this is the first prototype of acoustic dispensing invented by nature. Luckily, Labcyte got the idea and went ahead and developed some tools that we use in our everyday operations. So what FIMM is? It's a research institute operating under the University of Helsinki. We are about 200 people, 20 nationalities. We have a very strong molecular medicine research at FIMM, many different research groups. We have technology platforms, genomics, metabolomics, imaging, biomarkers. Our unit also is in the technology center. A new core unit to be included is single cell analytics. We also host some biobank infrastructures. And all of these entities work together to translate the research and hope to help the patients. So what our unit is doing, we have chemical and RNAi libraries. We share chemicals with academic groups. We do high throughput imaging, ultra miniaturized assays, but actually about 80 to 90% of our activities is concentrated on drug sensitivity and resistance testing, and we do this with leukemic patient cells. But equipment and libraries, we have some Labcyte equipment that we're quite happy with. So here is our Access workstation. It's integrated with two echoes, Kermit, which is a 550, and Miss Piggy 525. They are happily married in the Access. We have a DRAC LX bulk filler there. There is the regular sealers, peelers, centrifuges. The newest addition is a Saitama 10 plate hotel, which increases the plate capacity to well over 200 plates. We also have the oldest unit, the standalone FOSI. Then some other equipment. This setup was actually initially built by Jussi Taipalai's group, and it has in the middle an industrial robotic arm which moves the plates from one station to the next. It has a plate reader, cell incubator, all the regular stuff. We can also wheel in biotech EL406 washer dispenser. We also have two biotech multi-flow FX dispensers and two other plate readers, a Citation 5, another Ferrostar FS. And then in the institute we have a light cycler and some other equipment, some of which are shown here. We have two flow cytometers from IntelliCyte, IQ Screener HD and IQ Screener Plus. Then we have a high content imaging system, and we are about to purchase another scanning disk microscope system in the spring or early next year. Then we have an array printer and 3 IncuCyte live cell imagers. About the libraries, we have a genomic ambient silencer select siRNA library that we can use for whole genome screening, but we can also use the subsets or do cherry picking based on the needs of the users. The users often also provide custom libraries that we can screen for them. Chemicals, we have quite a big haystack to select from. The majority of the screening, though, is concentrated on the drug sensitivity and resistance testing, as I mentioned, and currently this library has 525 compounds. This is some details about the set. So we think it's currently the most complete clinical oncology collection in the world. About 1/3 of the compounds are approved, then there is plenty of investigational compounds and probes. And about half of the library is kinase inhibitors. There are also conventional chemotherapeutics and epigenetic modifiers, etc. It's fine-tuned. Usually the concentration is from 1 to 10,000 nanomolar, so we cover a 10,000-fold concentration range. But if the concentration is too high or too low, we adjust it according to the results that we see. So every new version of the library has more fine-tuned concentrations. Currently, the set consists of 8 380 well plates, 384 well plates, and in the next version we're probably going to drop out some compounds which haven't shown much activity and replace those with some new compounds. The library is quality controlled. If there is any doubt of stability of the compounds, we mass spec those, and we also check the compounds every 2 months using 4 control cell lines. This is just to show that the results are repeatable, so in run 1 and run 2 the correlation is very high. This set is available for the scientific community, so if you're interested, just contact us and we can send you assay-ready plates so you can use them in your laboratory. We can also provide you with custom sets and combinations. Then about screening and assays. This is just a flow chart of the operation, how it goes in this case in siRNA screening. So we start with the siRNA library. We use the echo to transfer the compounds into the assay plates. These can be kept in the freezer for years and they stay in good condition. Once they are needed, we add the transfection reagent and the cells with the bulk dispenser, incubate the cells either in a regular incubator or live cell imaging equipment. Then we can fix the cells, image them, or we can use other detection reagents and use the plate reader. In the case of drug screening, we naturally also use the echoes. We just transfer the compounds into the assay plates, keep them in storage pods under nitrogen gas, where they stay good for a month or 2, and we have them in stock. So whenever somebody needs those sets, we can give those sets to them. Jump back to the equipment. Previously we used multi-drop dispensers to add the cells to the plates. That had a problem in the sense that you have to use quite a lot of volume to prime the equipment, and still you might see some striping effects in the left part of the plate where you start the dispensing. So we acquired a multi-flow FX, and actually we see better consistency in the plate, and much less material is used for the priming. The cons for this is it's a little bit slower, but we can use a stacker, so it's no problem. And the cassette tubing wears out, but that can also be replaced. So then an example of genomic siRNA screen. You can't see much, and that's because this is unpublished data. There's a manuscript in preparation. So we started with the full genomic library. The primary screen revealed about 400 hits, which are shown here in violet. Then these went for, so the primary screen was 3 siRNAs per gene, and those were pooled. In the secondary screen, we used 6 siRNAs per gene, and that was 400 genes. And then in the end, the validation in the research group is now concentrating on 4 genes. And hopefully we'll see a publication about this soon. Then another siRNA screen. This was based on SNP data in patients, and the research group acquired a custom siRNA library for those genes which were around those SNPs in the genome. And we combined the siRNA screening with the anthracycline treatment in a concentration range. And these are basal breast cancer cell lines. These are luminal breast cancer cell lines. The blue bar is the siRNA negative control, and the orange is gene X knockdown. And the higher the bar, the more sensitive the sample is towards the anthracycline. And this is also a manuscript that has been submitted, so hopefully we can provide more data about this soon. Then this was actually the first sRNA screen that we performed for a user. That took place some years ago, and the validations were quite extensive and took a long time, but finally we were able to publish this last year. So the group wanted to know what are the interacting partners for two bait proteins, and they had then went through databases to search for potential interacting partners, but also using mass spec data, and then based on those candidate genes, they acquired a custom library that we used first in the PC3 cell line, a prostate cancer cell line. Then looking at the similarity between the bait protein and the candidate proteins, the genes were ranked. The same thing was done with the non-cancerous prostate cell line, again ranking, and after that bioinformatics by checking the gene expression levels in prostate cancer tissue versus healthy tissue, and then summarizing the ranks using the rank relevance platform. The group went to validate the results and actually found in the top hits known interacting partners, but also new interacting partners, which then will enable them to continue with their research project. So the principle works. Then something about miniaturized assays. This is just a couple of assays that we've done with the echoes. So these reactions have been set up completely with acoustic dispensing using the Echo 525. Cell-based assays using the Echo 525 for quantitative real-time PCR. We are using 2 microliter reaction volumes here. I'll also slide about this next. Then we also did a miniaturized HTRF assay in 1536 format using an Epigenose kit from SysBio, and here we were able to miniaturize the assay from the recommended 20 microliter into a 1 microliter reaction. So the cost reduction was quite good. Yeah, this is the quantitative PCR. So it's a 2-step reaction where we first lyse the cells and synthesize cDNA, and in the next step perform the qPCR. And here are some statistics. The red values that you see are at the detection limit, so we disregarded those values. But in general, the standard deviations were very small, as well as the coefficients of variance. The standard curves were nice, and we were able to continue with the analysis of the target genes. With solid tumor samples, the problem is often that the tissue that we get is not big, and it might be very heterogeneous. So the challenge is to get enough sample to run a bigger screen, and thus there is the need for miniaturization. And as I explained already, the conventional peristaltic dispensing requires considerable priming. So we have also started to test the Echo 525 for cell dispensing. This was one of the first tests that we did in 2013, I think, where we used HeLa cells and just used the HeLa cells in its own media and dispensed 100 nanoliters per well, corresponding to 1,000 cells. And in the upper picture here, you can see the results when we dispensed from a polypropylene plate without any priming, so there's considerably less cells at the beginning. When we primed into this well here, the result was much better, and the CV was actually surprisingly good, and there was not much variation throughout the whole plate. We've continued this occasionally. This was actually an experiment done by Carl in our laboratory, where he used the histidines that was mentioned earlier as well to control for the buoyancy of the liquid. And here we used HDQP1 cells and transferred 1 microliter per well containing 300 cells. We incubated the cells overnight and detected the amount of ATP in the wells by using the cell data glow reagent. So this is the ECHO 525 result. This is the biotech multiflow FX result. It's pretty similar. The CVs are about 10% plus minus 3 in both cases. The ATP levels are pretty much the same, a little bit more in this case. So some of the cells might suffer from the echo dispensing, but it's quite good, and this is something that we want to continue developing. Then about the drug sensitivity and resistance testing, which is, like I said, the majority of the operations in our unit nowadays. So what we get is primary cells from leukemic patients. We get them at diagnosis, but also at relapses. Unfortunately, the patients first have to go through the standard chemotherapy before this whole DSRT can make an effect on their treatment. But whenever there is a relapsed sample based on the DSRT results and the molecular profiling, the clinicians who get all the results can then use the data to treat the patient with the compounds that seem to be effective ex vivo. They also, of course, know which drugs are not effective, so they can avoid using drugs that should not be used. The whole platform was published in 2013, and we are in preparation of the next manuscript. This is the team involved. We have the clinicians, many research groups at FIMM. And how does it go in practice? Well, first we make the assay plates using the Access system. When we have a sample, we add the cells on top of the compounds into the assay plates. We incubate for 3 days and then detect first the amount of dead cells using cell screen reagent, and then we detect the metabolic level of the cells using the cell data glow reagent. We feed the data into a pipeline which provides us with the dose response curves plus other matrix, and after that we compare the activity of the results to controls to understand better the biological side of the disease and to translate the results back to the patients and find which patient groups might be responding to certain kinds of drugs, but also to enable drug repositioning. How we assess the data? First, we calculate the drug sensitivity score. It's basically the area under the curve where a control would have a very small area. The AML patient in this case would have a bigger area. So you have a difference which enables us to calculate the selective drug sensitivity score. It's a single number that tells quite a lot about the drug response. It takes into account the IC50 or EC50 value, the slope, the maximal, minimal asymptote, the drug concentration. And when you compare the patient DSS value to the control, you get the selective value, which is often the most important number that is looked at by the clinicians. So this is again from the 2013 paper. So here are shown the selective DSS values for these drugs, and the patients here can be broadly divided into 5 different groups based on the drug responses. We have also the mutation data, and in some cases we can start to find some links. For example, the FLT3 inhibitors linked to FLT3 mutations, MML fusion linked to MEK inhibitor sensitivities, and p53 mutations are found in the most unresponsive samples. So some links between the phenotype and genotype are starting to emerge, but for most of the drugs there is still no clear link between these. The DSRT has been used quite a lot already, over 1,800 times for primary leukemia samples, for different solid tumors, for cell lines. It has been used by us, it has been used by many academic groups worldwide. There are probably close to 20 different publications already published around the DSRT, and we have ongoing collaborations with pharmaceutical companies using this technology. And as Mark yesterday mentioned, there is also a clinical trial ongoing around the findings regarding axitinib. So what's next? Currently we measure the viability and death. Obviously it provides a very generalized information about what's happening in the sample. We are aiming to use imaging much more in the future, but also flow cytometry, where we actually have quite interesting data. We can separate different cell types in the leukemic samples and see that the different cell types respond differently to the compounds. Unfortunately now I don't have any slides about this. So the echo is instrumental here as well in basically everything we can use. If we use the cell titer-glow reagent which lyses the cells, we can use the array maker software to print the lysates into slides, and then use, for example, RPPA to detect different proteins. We can also make custom combinations about the compounds using the echo to test for synergies. And just to conclude the DSRT, we can group the disease based on the ex vivo drug sensitivities and link some of the drug responses to the genetic mutations. We can also identify actionable drugs and molecular drivers and then use the data to treat the patients. This is one of the most famous examples that we have, where a patient had been diagnosed with AML, then relapsed twice using the standard chemotherapy. Then a triple drug combination was applied, dasatinib, sunitinib, and temsirolimus. Unfortunately, there were serious side effects, so sunitinib had to be taken out from the care, and finally the patient relapsed again. But before the DSRT based treatment started, the patient had been in intensive care for a long time, and in about a week the patient was able to return home. So there are some good initial results, but no major breakthrough yet. Then something about data management. We have basically two different data management systems: commercial software from Dotmatics, which we keep to which we use to keep our compound registry in, also to analyze some of the data. We have also built an automated analysis pipeline called Breeze, and in this we host different analysis pipelines for DSRT, for combination screening, etc. So what is Breeze? It's a web interface where the bioinformaticians can put their scripts to be used by the researchers. It's really quite easy to just start a script which analyzes your data. And the data is also fed back to a database where in the institute-wide manner the people doing the research can assess the data. We're in the process of also testing this whole platform in the cloud environment, and we aim to be able to offer the BRIES tools to researchers outside of FIMM as well. So what it does, we have the data from the DSRT. It's fed into the BRIES system, which analyzes the QC parameters, does the curve fitting, calculates the DSS values, and feeds the data into a database. And then in the individual system medicine research programs, which currently is dealing with these kinds of samples, then the data can be analyzed in many ways. This is just to highlight another tool that we have in BRIES. It's called Synergy Finder. And here we can easily analyze data from a custom matrix plate, which has 6 different drug combinations in one 384-well plate. And this is just screenshots from the interactive program. So in summary, what the acoustic liquid handling has enabled us to do is preparation of the assay plates, which is very crucial for us. We can deal with chemicals, we can deal with RNAi. It's very efficient, accurate, and precise. We can easily do the dose response testing plates, both for the standard DSRT but also for custom combinations. And we've also seen that it can be used to dispense cell lysates for follow-up assays and also live cells. And this is our unit at the moment. Christer Wennerberg is the head of the unit. We have 2 senior researchers in the unit, and Evgeny Kuleski is our assay developer. Then our robotic and chemical wizard, Laura Turunen. Here, many of you have seen her. We have skilled technicians and 2 bioinformaticians in the unit. And for more information, you can check these articles or you can email me. Thank you very much.